So today we're going to talk about microbial metabolism, okay? So this is really important because we've talked all about microbes, but let's learn about what makes them tick. Okay, so looking into this, you can see that this is one of the overviews, is scoping out the chapters, right? And so then that is a big old picture, and it says that this is the metabolic pathways, and it's guiding the flow of reactions down the right track. And so basically, what is metabolism? Metabolism is all chemical and physical workings of a cell. And there are two types of chemical reaction. There's catabolic and anabolic reactions. So catabolic is a breaking down reaction, where anabolic is a building up. So I say I have a cat named Sophie, and she likes to break things. Now, really, she doesn't. It's a fun little kind of thing. But anyway, you can remember that catabolic is breaking down. So catabolic or catabolism is a degradative reaction, and this breaks the bonds of larger molecules by forming smaller molecules, thereby releasing energy when we're specifically talking about ATP. And then anabolism is those anabolic. Okay, think of back in the day with anabolic steroids. People used to use them to bulk up. So these are processes that are used in biosynthesis. This is processes that form larger macromolecules from smaller micromolecules, and this is going to require the input of energy. So looking at these, we have ways to make or break, okay, reactions. So when we're looking at a synthesis or a condensation reaction, this is an anabolic or building reaction that forms covalent bonds between smaller substrate molecules. This again is going to require ATPs, molecules, and it's going to release one molecule of water for each bond that is formed. So the synthesis, this is like also like a dehydration reaction, right? So when we're sticking things together, we're making them like beef jerky. We're making them stick closer together. So because of that, we're removing a water, okay? So that would be a condensation or condensing type reaction, okay? And so that would be that guy. Where hydrolysis, we're using hydro or water to lyse or break, right? So a hydrolysis reaction then would be that catabolic reaction that breaks down substances into smaller molecules, and this is going to require the input of water to break those bonds. So why do we care? Well, we care because, we do a lot of functions in our body, and one of them is going to be actually a lot of those reactions are catalyzed or helped out by the use of enzymes. Well, enzymes are biologic catalysts that increase the rate of a chemical reaction by lowering the energy of activation, which is the resistance to reaction or the activation energy. The enzyme is not permanently altered in this reaction, and the enzyme promotes a reaction by serving as a physical site for the specific substrate molecules to position. So substrate is a fancy word for starting material. They both start with S's, okay? So remember that enzymes make things quicker and easier. So I kind of joke and say that this is like if I'm getting out of bed in the morning, normally it'll take me 5 cups of coffee, but with an enzyme, it would only take me like 2. So it's going to be quicker and easier for me to get out of bed through the use of enzymatic reactions. Okay, so you can see here, this is one of the stereotypic pictures that they always use for enzymatic reactions. I'm trying to find it in your book. So this is on page, in mine, it's on page 225. This is figure 8.1. And so you can see that this is the progression of the reaction. Here, let me move that down just a hot minute. Okay, so this is how the reaction is progressing. And you can see that the green line is with an enzyme, the blue line is without. So this is how much energy it takes to do that reaction. So without an enzyme, it's going to take this much energy to do a reaction. But with an enzyme, since things are becoming quicker and easier, it takes less energy. And this area right here is called activation energy. So you can see that the activation energy with an enzyme is significantly reduced. Okay. So moving on to, well, what are enzymes? So let's look at that enzymatic structure. And this is on figure 8.2. So simple enzymes consist of just the protein alone, and conjugated enzymes or holoenzymes, these contain proteins and non-protein molecules. The apoenzyme, this is a protein portion. The cofactors are non-protein portions. And these include like your metabolic cofactors such as iron, copper, and magnesium. And you also have coenzymes, and these are kind of like helper enzymes. And these are going to be organic molecules, and some of them can be vitamins. Okay, so again, this picture here is showing a conjugated enzyme structure. And again, this is figure 8.2. And all examples have an apropet enzyme, which is a polypeptide or a you can see right there, or a protein component, and one or more cofactors. Okay. So when we're looking at types of enzymes and their cofactors, then this is a fun little summary table. And this is table 8.2 as it says right here. So you have a catalase, and this is going to break down or catabolize my cat Sophie likes to break things. Catalyze or break down, right? And then a metallic cofactor that's required would be iron. Oxidize, this is going to be adding electrons to an oxygen, and your metallic cofactors would be like iron and copper, hexokinase, urease, nitrate reductase, and DNA polymerase. So a fun thing that's been mentioned before, and I'm sure it's going to be coming up here too, is all enzymes tell you they're enzymes because they end their name in ase. So if you see all of these enzymes end in the word ase, or end in the suffix ase. So that will tell you that it is an enzyme. And so all enzymes are proteins. Not all proteins are enzymes, okay? But enzymes are specialized proteins that end in the suffix of ase. So when we're talking about a temporary enzyme substrate union, let me move my picture out of the way, okay? This occurs when the substrate moves into the active site, and this is called an induced fit. And I kind of think of this as like a little snuggle. And so when the enzyme and the substrate come together, the enzyme's like, yay, I get you. I'm going to just, snuggle you just a little bit. And this is induced fit. And so they appropriate, or the appropriate reaction is going to occur then. And then the product is formed and released. Okay. And so this would be an example, this is figure 8.4 of an enzyme substrate reaction. So in A, when the enzyme and substrate come together, you can see here, the substrate, which is that S, right? Fancy word, substrate, starting material, will have the correct fit and position with respect to the enzyme E. Where in B, we now have them bound together. And so this is the enzyme substrate complex, because it's a complex because they're together. So when the enzyme substrate complex is formed, it enters into the transition state. And during this temporary but tight interlocking union, the enzyme participates directly in breaking or making those bonds. So then we move to C. And in C, once the reaction is complete, the enzyme is going to release that product. Then we go to D, and the induced fit model proposes that the enzyme recognizes the substrate and it adapts slightly. Okay, so this is just showing you that induced fit and where it snuggles. It's going to adapt slightly, and then it really gives a good little snuggle so that the final binding is even more precise. Okay, so you have your enzyme and your substrate, and then they become the enzyme substrate complex, and then they actually have an enzyme product complex, and then you release the products when you are done. But the takeaway from this one is induced fit. It's a little snuggle, right? It's like a key or hand and glove, key and lock. It fits very tightly, so it's a very specific reaction that is occurring. So looking at those cofactors again, well, cofactors are going to help support the work of these enzymes, okay? So this is figure 8.5, and this is the carrier functions of coenzymes. And many enzymes use coenzymes to transfer those chemical groups from one substrate to another. So in A here, you can see that the enzyme with the coenzyme is positioned to react with two different substrates, okay? Then the coenzyme picks up a chemical group from substrate 1, and then the coenzyme readies the chemical group to transfer the substrate to the second, okay? And then the final action is to group, or for that group to be bound to the substrate. Then two altered substrates are then released from the enzyme, okay? So again, micronutrients are needed as cofactors, and they are just assisters, okay? They're assistants. So where's all this happening? Okay, well, this is in figure 8.6, and these are the types of enzymes as described by their location and actions. Okay, so we have exoenzymes and endoenzymes. Look for those keywords. Exo means outside of, endo means inside of. So those exoenzymes are going to be transported extracellularly or outside the cell, where they break down large food molecules or harmful chemicals. And these would be like cellulose, amylase, cellulase, amylase, or penicillase. Okay. And so those endoenzymes then, the ones that are inside, these are retained intracellularly and they function there inside the cell. And most enzymes are endo, okay? They're going to be functioning within the cell. So how do we regulate these? Well, we have constitutive enzymes and we have regulated enzymes, okay? And so this is figure 8.7 in your book. So constitutive enzymes, these are always present. They're always produced in equal amounts and they're at equal rates, and then it's regardless of the amount of substrate. Where regulated enzymes is going to be, they're not constantly present. The production is turned off, which would be induced, or I'm sorry, turned on, which would be induced, like kind of like induced labor, we're turning on labor. Or they can be reduced, which is repressed. I have repressed memories from childhood. I really don't, but repressed is turned off, right? In response to changes in the substrate concentration. Okay, so you can see here that this is the constitutive, okay, where this one is we can add more substrate, remove substrate, and we can affect the level of the enzyme. So what about the sensitivity of enzymes to their environment? Well, the activity of an enzyme is influenced by the cell's environment. And enzymes operate under temperature, pH, and osmotic pressures of the organism's habitat. Okay? So when the enzymes are subjected to changes in the organism's habitat, they become unstable. You get them out of their happy zone, they're not going to be very happy. So we have two terms, labile, and this is where we have a chemically unstable enzyme, or denaturization or denaturing. And this is weak bonds that maintain the shape of the apoenzyme are broken. And so they change their shape. Shape is all for function here in enzymes. When you change the shape, they're not going to function well at all. So how do we regulate this then? Well, this image here is in figure 8.9, and this is the patterns of metabolic pathways. So in general, metabolic pathways consist of a linked series of individual chemical reactions that produce intermediary metabolites, and they lead to a final product. These pathways occur in several different patterns, including linear, cyclic, and branched. And you can see you have linear, cyclic, or branched, either divergent or convergent. So anabolic pathways, so anabolic are those building pathways that are involved in the biosynthesis, result in a more complex molecule where each step adds on a functional group. Whereas a catabolic, or the breaking down reactions, involve the dismantling or breaking down of molecules, and this can generate energy. And virtually every reaction in a series involves a specific enzyme. So enzymes are specific. They only catalyze or do one reaction. They can be reused, but they're only specific for one reaction. So here then, what are the direct controls on the actions of those enzymes? Well, we have competitive or non-competitive inhibition. So inhibit means to stop. So if you're competing, well, then you're competing with that active site. Where non-competitive inhibition, you're still competing, but it's just not at the active site. So competitive inhibition, this is where the substance that resembles a normal substrate competes with the substrate for the active site. So I kind of explain this as a door and a key. So the wrong key will fit into that door lock, but it's not going to open the door. Okay, so this is competing at that active site, but it's not going to work, but it's blocking it, right? It's like just competing with it. Where non-competitive, this is where it's like, dude, I'm not even gonna mess with the front door or the active site. I'm busting in through this alley or this allosteric site. So these are where the enzymes are regulated by the binding of molecules other than the substrate away from the active site. So not at the front door, through that alley or that allosteric site. And two of these would be enzyme repression. This is where it inhibits at the genetic level by controlling the synthesis of key enzymes, or enzyme induction. And this is where enzymes are made only when suitable substrates are present. Okay. So what is this then? The regulation of the enzyme action. This guy here on the left, this is figure 8.10, and this would be the competitive inhibition. Okay. And so you can see that the normal substrate and the competitive inhibitor, they're very similar shape. Think of those keys, right? Both molecules are going to compete for that active site. When the actual substrate or starting material binds with the active site, then we're going to have that reaction that is going to happen or proceed. Where if it doesn't, because it's blocked by a similarly shaped competitor, it's going to inhibit and therefore is going to be incapable of making the product. Where the non-competitive, again, we have that substrate and we have the active site, but we have that site that's distinct from the active site. So normally what happens is, yeah, we would go ahead, we'd bind, and we'd have that reaction occur, and we'd make that product. But then when we do this allosteric, if you follow the same thing of like busting through the alley, well, we're going to have to get a contractor in to fix the problem, right? And so it's going to take more time, and it's going to be a little bit longer lasting, and it's changing the shape of your house, or in this case, the enzyme. So you can see that the substrate shape is no longer the same. So when we bind to that allosteric site, it changes the shape of the active site. So now the substrate can't bind. And so then this reaction is blocked because the binding of the regulatory molecule and the regulatory site changes the conformation, fancy word for shape, of the active site so that the substrate cannot enter. So what about enzyme repression? Well, this is figure 8.11, and this is one type of genetic control of enzyme synthesis, and this is called enzyme repression. So in steps 1 through 5, you can see that the genetic controls are active, and then the enzyme is synthesized continuously until enough product has been made. But then down here in 6 and 7, you can see that the excess product reacts with a site on the DNA that regulates the enzyme's synthesis or the making of that enzyme, and this thereby inhibits further enzyme production. And this is like feedback inhibition. Okay, enzyme repression, feedback inhibition, kind of like the same thing. Okay, but basically your body's responding to, hey, we have enough product, let's shut it down production here temporarily. Okay. So what about the pursuit and utilization of this energy? Well, what is energy? Energy, by definition, is the capacity to do work or to cause change. And forms of energy include thermal, radiant, electrical, mechanical, atomic, or chemical reactions. And so when we look at cell energies, well, the cells manage energy in the form of chemical reactions that either make bonds or break bonds, and they transfer the electrons. So we have endodermic reactions, and these are the ones that consume energy. So you have the energy plus A and B, you have the action of the enzyme, and it's going to produce a product C. Well, then we have exergenic, exer, right? So that's going to be releasing energy. And so then you have X plus Y with the action of the enzyme, it forms Z plus energy. And the energy released is temporarily stored in high energy phosphate molecules. And the energies of these molecules is used in the endodermic cell reactions. Okay. So if we look at this figure, this would be a picture of cellular energy processes. Okay. So you can see in, step 1 that the glucose is going to be oxidized as it passes through the subsequent metabolic pathways, resulting in the removal of the hydrogens and their accompanying electrons. And then during this part of the pathways, the glucose carbon skeleton is also taken apart or dismantled, giving rise to the end product CO2. These reactions lower the available energy in each successive reaction, but they effectively route that energy into useful cell activities. And then here in 3, the energy in electrons and hydrogens is captured and it's transferred to the ATP. And ATP is the currency of our cell, right? It's spent to drive the thousands of cellular functions that require energy. And then in 4, in aerobic metabolism, aerobic is with oxygen, the electrons and the hydrogen ions are generated by the respiratory pathways, and they combine with oxygen to produce another end product, which is water. Okay, so that would be your end products, and that's actually a offshoot of the electron transport chain. Okay, so that would be the cell energy processes. Okay.